Hello, I am Haley A. Clark, and I will be presenting my poster on microwave-assisted organic synthesis and characterization of drugs, solubilization studies of drugs in aqueous media using cyclodextrin. Cyclodextrins are ring-shaped oligosaccharides that are used for the improvement of solubility and are produced by enzymatic conversion of starch. The pharmaceutical industry uses cyclodextrin because it serves as a powerful delivery agent. Cyclodextrin is available in three forms: alpha, beta, and gamma. The difference in these forms is the number of glucose monomers. Alpha has 6 glucose monomers, beta has 7 glucose monomers, and gamma has 8 glucose monomers. 2-hydroxypropyl gamma cyclodextrin is a gamma cyclodextrin byproduct of an 8 sugar ring molecule. 2-hydroxy gamma cyclodextrin is used to increase solubilization of macromolecules and cell culture applications. Unlike 2-hydroxypropyl beta cyclodextrin, 2-hydroxypropyl gamma cyclodextrin can work with larger molecules. 2-hydroxypropyl beta cyclodextrin is the most widely used modified cyclodextrin. It has 7 large glucose units, low toxicity, and cholesterol binding properties. We found that we can dissolve 2-hydroxypropyl gamma cyclodextrin in aqueous media while heating and stirring, then adding a drug dissolved in DMSO. This solution and the drug goes into the solution. This drug is now soluble in the media, and we can test the efficiency of the drugs killing cancer cells, specifically prostate and breast cancer cells. The following were mixed in 2 milliliters of dimethylaniline, 2 milliliters of methylstyrene, and 0.2 milliliters of trophyl methane sulfuric acid in a 10 milliliter reaction vial with a stirring bar placed inside. We placed this reaction vial in the microwave synthesis reactor at a power of 7. We heated it to 250 degrees Celsius with a 600 RPM stirring speed. This reaction vial will be held at 250 degrees Celsius for 6 minutes, and it will have a 1-minute ramp time. After heating, the vial should cool to 70 degrees Celsius. We then ran an NMR analysis. The collection of the product, the dissolved reaction mixture. We dissolved the reaction mixture in 10 milliliters of ethyl acetate in an aluminum flask, and we added it to a separator funnel. We rinsed the flask with 5 milliliters of ethyl acetate and added it to the separator funnel. We performed this step twice to ensure that all residue was gathered. This step, the next step was done twice also. We added 20 milliliters of sodium bicarbonate and into the separator funnel. We put the stopper in place and gently shook it to ensure that everything was mixed and no residue on the side was left. We removed the stopper and proceeded to remove the sodium bicarbonate. We added the product left in the separator funnel to the aluminum flask and added 3 grams of magnesium sulfate. We filtered the product and let it dry for about 14 hours. So in this discovery and application of the new drug, there is often a problem of getting the drug delivered to the application site. We are synthesizing novel drugs to test the efficiency of them on killing breast and prostate cancer cells. The drugs are insoluble in the media that these tests are carried out in. We decided to study the use of the cyclodextrins for solubilization of our drugs in media. Cyclodextrins are ring-shaped oligosaccharides that are used for the improvement of solubility and are produced by enzymatic conversions of starch. The pharmaceutical industry uses them because of this. The objectives that I studied was to discover the means to solubilize our drugs in aqueous media that is used to study effectiveness of drugs killing breast and prostate cancer cells. Cyclodextrins are produced by enzymatic conversion of starch. We found that we can dissolve 2-hydroxypropyl gamma cyclodextrin in aqueous media while heating and stirring, and then adding the drug dissolved in DMSO to this solution. And the drug then goes into this solution. The drug is now soluble in the media, and we can test the efficiency of the drugs killing both prostate and breast cancer cells. If you look at the picture here under results, The first picture shows the drug that did not go into solution. The cyclodextrin that we used was 2-hydroxypropyl beta cyclodextrin, and then the vial on the left holds a vial with when the drug did go into solution after heating and stirring. The cyclodextrin that we chose to use was 2-hydroxypropyl gamma cyclodextrin, which led to our conclusion.